what kind of links them together is the sort of the application and the purpose, the general purpose, which is sort of understanding this kind of transfer of information from the signals and the conditions that we get around the cell, so in the environment of the cells, which sells sort of different information into the cell, which then is translated into different interactions that are happening within the cell, between different biological systems, and then this turns into decisions, actions that the cells take to, say, differentiate, to fight a virus with an immune system, or divide, and so on. So the two parts are quite different in terms of approach and methodology. One of them is with mechanistic modeling and talking about the dynamics and the interactions of molecular populations that lead to these dynamics, and how do we simulate from it, analyze them, and feed them to time series data. The second part is more talking about quantifying the information transfer and how this is translated into decisions, and kind of more approach go directly to data first and learn from the data on that part. Now, one last thing before I move on. So I'll be talking about cell environment and cell signals and conditions and molecular populations and then decisions of cells. But feel free to replace that in your mind with another setting. For example, I'm aware in epidemiology, we talk, instead of conditions, we might talk about interventions, in terms of signals interventions. In terms of, instead of molecular population, we might be talking about compartments of the model, say SIR, say SIR models. And then actions may be the actions of individuals or populations and so on. So yeah, so in the first part, I'll be talking about stochastic simulation and inference for biological systems. The outline is, I'm going to talk about some motivating examples for the approach that we follow, then talk about the models that we use, introduce them, and so on, and then talk about inference for them in terms of parameter estimation and so on. So the first motivating example is the circadian or 24-hour clock that operates in our cells and in most living organisms, and seems to be regulating things like when we go to sleep, when we are hungry, and lots of other things that happen in our body. And it seems to be, the disruption of it seems to be having implications in health. Yeah, and during the last years has been understanding a bit more on the importance of the clock, and we have fields like chronotherapeutics and so on emerging during the last years. Yeah, growing a lot in the last years. So there's lots of things we want to know about the clock, but we do know that they present oscillations. So they have these cycles, which are roughly 24 hours, that they involve multiple genes, and they are interacting between themselves in kind of non-linear ways and complex interactions. Yeah, and that the data are quite stochastic. They are quite noisy, and we want to understand this stochasticity. Now, the other system that we're studying is the NF-kappa-B system. This is a signaling system that responds to various signals, including those that are related to inflammation. So when we have inflammation in our body, NF-kappa-B is a system that senses that and kind of triggers responses like turning on the immune system and so on. So, and again, the general characteristics are very similar. It also has this periodic behavior, about 100 to 200 minutes oscillations typically, involves multiple genes and their multiple kind of molecules they're interacting, and it's quite stochastic, it's very noisy and so on. So the general target is for this kind of models to try to build models and also simulation algorithms and inference algorithms that would be accurate, so it would be giving a good description of what's happening in the cell, but also at the same time being computationally fast, do inference with them, and that they can apply to these sort of large systems that we're studying. Yeah, so that's the general purpose of this. So I'm gonna try to give a 1 or 2 minutes introduction to reaction networks, which I'm not sure you are familiar with them, but I'm sure you've seen quite a lot of presentation of SIR type of models. So it's more or less the same thing, but still I'll introduce them a bit. So this is a diagram of that. And in the bubbles, these colored bubbles, we have the different molecular species that they're involved, the different molecular presenting molecular populations. For example, this is the pair mRNA. We have 2 genes in here, pair and team, like period of time. And yeah, they are in different forms, presenting different forms in this system. So these are the species in the bubbles, and then the molecular populations, in other words. The arrows are the transitions that are happening, and they're happening because of molecular, biochemical reactions, but mathematically they are state jumps. They have jumps of our states from one thing to the other. For example, this one will say that the pair mRNA is translated into protein, and we get a pair protein molecule, from a mRNA molecule. So what are we recording? We're recording the molecular population, so the number of copies of each of those species in this exercise. And then the jumps we have them like that. So we're saying, okay, if the state was X and the reaction of the J type happened, then the state becomes X plus new J. For instance, in this case, we're losing 1 pair mRNA molecule, gain 1 pair protein molecule. So you have this change here, you have -1 of the first population, +1 in the third population, all the others are 0. So then the times for each reaction, how the waiting times, under some biochemical assumptions about how the cell environment is, we can model them as exponentially distributed times. And this leads us to a Poisson jump process in continuous time, which we can describe in different ways. We have this random time change representation, or we have this so-called chemical master or master equation, which are basically the forward equation, and they describe the evolution of this transition probe. They're moving from X0 state to X and so on. Now, there is one characteristic I want to talk about, these rates. So the rates of exponential times, they depend on the current state, right? The dependence is depending on the type of reaction that you have. Some of them are linear functions, some of them non-linear functions, like this one. But the key thing is that they depend on the current state, which means that we have inhomogeneity in terms of time. So it's not constant rate Poisson processes, one that changes, the rates change over time. Now, this is what complicates things, because it means that we very rarely can solve this master equation, which in a more practical terms means that we have no analytical expression for the transition probabilities. So there's not much to do with this master equation other than seeing the form of it, essentially. So then, can we simulate from it? Yes, we can simulate quite easily. It's very easy to implement those exponential generation, exponential times, and so on. And this is exact simulation, so you can exactly simulate from that equation, but it's very slow because it generates every single reaction that happens, it will generate it there, so all the reactions. So that becomes very slow when you have even a moderately complicated model. So from this, you have a whole branch of maths and so on that they focus on making this faster and still remaining accurate. So compared to that, this exact sort of simulation to be able to have the same kind of distributions that you get from this, but in a much faster way. So there's a bunch of them. I'm going to skip some of them, save some time, and I'm going to focus on this one called the linear noise approximation. The general idea is, the kind of way that you're getting it is using some sort of central limit theorems and some sort of Taylor expansions on the master equation, and you end up with this fairly simple form. So the state of the system is this linear combination of two things. phi t is a solution of the classical ODE system. So if you have a standard system, then you probably write it as an ODE, say the SIR kind of classical ODEs. That will be the phi t there. And then you add this psi t, which is a solution of an SDE, stochastic differential equation, which describes essentially the perturbations of the system around this deterministic solution. So you allow the sort of noise dynamics to go in the SDE, right? Now, and then you have this omega, which is in kind of this molecular setting, would be the cell volume or something related to the cell volume. More generally, you can think of it as something that is inversely proportional to the variance. You have larger omega, smaller variance, and vice versa. Yeah, so you can fix it somehow. You can find it so that the general noise you have in the system is described by this parameter. Right now, why is focusing on that? Because this SDE can be solved. You can find a solution of that, which means you have transition probabilities for it. And so, you know, if you want to get likelihood of time series, then it's possible with this one. So it's, you know, all these things that we want to do are possible. They're quite fast. The trouble is that this is an approximation which works in limited cases, which are still important, and we take advantage of those. It works well when you have relatively short transitions. So when you start from a point and then you don't want to look very far ahead, but some relatively short steps, then it's very accurate. When the dynamics are such that the dynamics are essentially linear and stable. For example, if you have sort of fixed point for the ODE, like this one, stable point, then for long times the LNA would be describing them. And yeah, more generally, if you have linear and stable dynamics. If you have nonlinear dynamics, because of this Taylor expansion that go on into the approximation, it fails. For example, I'm just showing you here that it fails. These are the SSA simulations, and this is the LNA contours, the distribution of the LNA, and just fails to approximate them. So we went back and tried to understand why it fails. And by looking at just the theory of dynamical systems, deterministic dynamical systems, we saw that if you have a periodic solution, which gives, this is sort of the graph of this periodic solution, we call them limit cycles. If you're familiar with that, then and you look at any point on this limit cycle, that's the phase of this periodic system, and you take a transversal section of it, then the dynamics there on the transversal sections, which are, bear in mind, if you have an n-dimensional system, this is n-1 dimension, you just lose one dimension, yeah? So the dynamics there are linear and stable. And if you take now the stochastic simulations of an oscillator, and you find these points in this kind of approximate way, you choose a section and you collect all the points when crossing from there, and you draw the empirical distributions, compare them with the analytically derived LNA distributions, you see that the LNA, so these are conditional LNA distributions, and you compare them, you see that they match very well. So when the dynamics are linear, kind of seeing that when the dynamics are linear and stable, we get the LNA to work. Yeah, LNA is a good approximation. So why it fails is because then dynamics on the other dimension, the tangential direction, so this goes along the limit cycle, which then this is related to the phase and the period of the system. In that dimension, the dynamics are not stable. Yeah. So what we see, this is translated in the stochastic dynamics. If we compare the period of the deterministic system with the period that you get, so the time that you need to do one cycle with a stochastic system, if you compare these two things, then and take the difference, and the variance of this difference, which we call phase deviation, is increasing linearly in time. Now it's increasing, meaning that it breaks down the whole approximation. That's why you get this failure. But on the other hand, the fact that it's kind of well-behaved and linearly increasing means to us that you could control it by kind of resetting the phase in regular times. So that's what we do. For example, in terms of simulation, what we are saying is, okay, so initialize the system just like you would do in the standard LNA, have some initial condition for the ODE, SDE, and so on, do a standard LNA step. And if you saw that the state moved too fast compared to what you expect with a deterministic system, then find the closest point according to some distance measure to the limit cycle solution. So bring your initial condition of the deterministic system to your new initial condition here instead of that. Correct your noise to be this one instead of what we have here. So the noise, the perturbation is only on the transversal section, which is well controlled, and move to the next step. So adding this extra step, this what we call phase correction step into the standard LNA seems to be working well. It's, you know, so we compare the distributions of SSA, the stochastic simulation, with LNA stochastic simulation, and they match almost perfectly. There's nothing to see that they're different. And it's way much faster. So it's hundreds to thousands of times faster. Instead of reading an HPC to do that, you can run it on your laptop and get in a minute or so, get some number of simulations. And we compare also to the standard competitors, say, other methods that you use to speed up the SSA, and it's still much faster than them. You see the times that they need. You need kind of half a second for something that one needs 11 seconds or so on. So that was good. Then what else we can do with that? So we want to feed it to time series data. So we could apply a sort of measurement equation on the state of the system. So this is some standard multivariate normal noise. It's just measurement noise. And this B is a matrix which could be either removing some entries of the state. So say we don't observe all the variables, or maybe we observe some function of the variables, some linear function of the variables. Right, and we have this kind of data and want to apply it on them. So you can work out the likelihood by deriving. So we derive the Kalman filter that gives you the likelihood of time series under the PCLNA model, and it's fairly standard compared to the. So it's very similar to the LNA, the standard LNA Kalman filter, apart from these phase corrections that we have another step there. So it's not too bad computing it. I mean, it's still a computationally heavy thing, but it's doable, which is very unlike what we have in this setting. Yeah, with this large system, it's very rare that you can compute likelihood of time series observations with these underlying mechanistic models. Yeah, and the likelihood function is a product of multivariate Gaussian PDFs. So we tried different MCMC methods on it, and it seems to be behaving nicely. We use mostly parallel tempering MCMC. It seems to be behaving a bit better than the others without doing a full comparison, to be fair. Now, the thing is, what we cannot throw under the carpet is that we have lots of parameters in those models. So for instance, these two models, we have 40 or 30 parameters to estimate. They are all related to each other, might be like of birth of a gene and death of a gene. So they're very related. So you have a big identifiability issue. So before jumping into finding the best MCMC algorithm that works better, we're saying that we need to see first whether the likelihood changes when you change a parameter. Yeah. So there is information there to estimate the parameters. So we compute the Fisher information matrix for that, which gives you the sort of the sensitivity to the different parameters. And we perform some analysis on the Fisher information matrix that kind of allows us to predict which parameters we can estimate. Yeah. Now this is feasible because PCLNA has this Gaussian underlying distribution that we can compute. You can't compute on the Kalman filter, so you get some sort of observed information matrix. What we did instead was something that is less computationally heavy, saying we can compute. So if we choose different phases of this limit cycle, and we can do as many as we want, so cover all the phases that we have, all the locations of this periodic system. And we compute the distributions of jumps from one location to another, and then going around into lots of locations. Then this gives us a big multivariate normal distribution. And if you have, so because we want to do Fisher information matrix, it's very rare that you have, some analytical expression to get a Fisher information. But when you have a multivariate normal, it has an analytical expression. You can sort of compute it by solving some of these, getting those, and you're done. So this is, this is easy to compute, fairly easy to compute. Yeah. So you can get, and we're asking whether this is enough to tell us about the sensitivity of our models. So we do different types of analysis. I won't spend all the time on that, but for instance, you're finding that the sensitivity when you have a stochastic model is much better than when you use a deterministic model. So you can estimate more parameters by accounting for the stochasticity in the model. You can ask experimental design questions, what time should I observe, what variables should I observe, and so on, using this tool. And in terms of estimation, so we have all these parameters. So we're starting and saying, okay, does this Fisher information matrix analysis gives us good predictions of what we can estimate? So if we see that the sensitivity to some parameter is high according to this analysis, does that mean that we'll be able to find, that the MCMC converges and gives us posterior distributions and so on, that seems to be learning from the data and so on, all these nice properties that we want for the MCMCs. It seems to be, if you take the most sensitive, then you estimate well, and we're starting adding some more parameters which are less sensitive and see whether we can estimate as well. And it seems to be, at least for the NF-kappa-B model, we have this nice relation that you see that the more sensitivity you have on a parameter, the smaller is the coefficient of variation. That's noise to signal, so standard deviation versus the mean of the posterior. So yeah, it seems like the sensitivity that we find in the Fisher information is reflected into the how accurate we are in estimating the parameters with a Bayesian inference. Right, and we applied that also for the circadian clock, which has a bit more sensitivity than the other model, and we estimate a bit more parameters. This one, kind of similar relation with in terms of the sensitivity and the coefficient of variation. So I'm not sure how we're doing time, but yeah, reached the end of the first part. Oh, I think I'm going quite fast, so please stop me. So let's summarize the first part. Right, biological data give rise to different challenge, methodological and computational. We want to do things fast, and want to do it, accurate and so on. And we deal with these non-trivial stochasticities and their complex interactions and so on. There are not many models that they are having all these things. So you can find the exact algorithm that is very accurate, but is slow. There are algorithms that they are faster, like the standard LNA, but is not so accurate in all cases. And the algorithm that we're proposing is not solving this, but is at least finding a solution for a specific class of system. The oscillatory system, I think, is finding a good enough solution that satisfies all these properties. Yeah, so we're looking to extend that obviously to other things. Yeah, and then in terms of inference, I think one key thing is, well, first of all, this model gives you a chance to do inference, whereas other models, it's very hard, impossible essentially for large systems, maybe very hard for smaller systems. And the other thing is that the Fisher information matrix analysis kind of rings a bell that, before we jumped, we could have jumped into the best MCMC and the best properties, but I suspect, as we will expect with all these parameters, we'll not be able to find good properties of the MCMC because there was no information for all the parameters there. So it's kind of ringing a bell that maybe we should look at sensitivity analysis before we dive into the best MCMC, choosing the best MCMC algorithm and so on. Okay, so that was the first part. Do you want to make any questions here or want to leave them for there?
That's up to you. You choose.
Maybe we should talk at some point. There are 2 or 3 or 4 concepts here which are very very close to the things that we've been thinking about.
We can
Not
keep-
for this kind of models, but not for this kind of application, but for another application. So the stochasticity, the value of information,
Mhm.
yeah, interesting. So I would have lots of questions, but
Yeah.
let's continue.
Yeah, that's very good. Right. So the second part, as I said, is more about the information transfer and kind of more diving into the data rather than, and and on the mechanistic models. So I'm going to talk about single cell RNA sequencing data and how the collaborators that produce the data that I'm working on, they kind of change the kind of purpose of this the typical experiment that you get there. How do we use them to quantify biological communications or signaling in biology, and then do some data analysis to see what we can predict with what genes, and also talk about these hierarchical and multifactorial classifiers that we use. So not sure you're familiar. That's a very broad picture. So I hope we have no biologists around to killing all the biology of this. But in my mind, at least, how it goes is you have a tissue which has lots of different cell types in it combined, and you kind of you don't know exactly what you have there. You just see a tissue, right? Then you isolate the individual cells, and then on each single cell you tag a biological barcode, I'll call it, on it, so you're able to identify the single cells. Then the DNA is cut in pieces and you do sequencing, so reading the sequences of DNA, and you put everything together in this big table, where, for example, the 7 here would say that we have 7 copies of gene 3 in cell 2, the second cell. And this is high dimension. This is the sample size. This is the number of genes are typically we start with, at least. So then what we do several pre-processing steps, normalization of data, filtering of genes, and so on. And then often we reduce the dimension using different methods like PTA, t-SNE, UMAP, and so on. And then we focus on clustering quite often to identify cell groups which might be representing actual cell types or subtypes, and the markers for those. So the genes that they're differentially expressed, we say, so they behave differently in one group than the other. So the idea is that, if you have a cancer tissue and you want to understand the complexity in it, the heterogeneity in it, the different types that you have, this is the first way that we had to do this, really, because we can identify the different cell types, see what is different in one cell type from the other, and so on. Now, we're not doing that. Doing something similar, though. We take a homogeneous mixture of cells. So if they come from a cell line, so in theory, they're all identical to each other, but then we split them in groups and we treat them with different conditions. So instead of trying to understand the heterogeneity in a heterogeneous cell mixture, you try to understand the effect of conditions on these identical cells. Yeah. And then the procedure is very similar, apart from statistical analysis, obviously, that I need to figure out and I'm working on. So for experimental design, we've got 8 groups of cells, so we split them in 8 groups. 4 of them are in normal temperature, 4 of them are in high temperature, 40 degrees. And then the other treatment is this TNF-alpha treatment, which is an agent for inflammation. It tells us that we have inflammation. And there are 4 different treatments. One of them is no treatment, no application of the agent. The second one is a 5-minute short pulse of TNF-alpha, and then following that with 200 minutes of no treatment, then 5 minutes again of another pulse. The other one is 5 minutes every 100 minutes instead. And then the last one is a continuous signal, TNF-alpha. So we have 2 different signals, I'll call them in this context, temperature and the agent. And the agent has this sort of, I call it hierarchical structure, because you're saying, is there an agent or not? If there is an agent, then is it a continuous treatment or a pulse treatment? If it's a pulse, then is it 100 minutes frequency or 200 minutes frequency? So I'll call that a hierarchy of the agent. Right, so what do we want to do for that? Again, it goes a bit back to what is signaling, what we care about in signaling. So the idea is that you have, the cells, they are in our bodies and they receive lots and lots of signals any given time. And sometimes, they will decide to fight, say, against the virus, turn on the immune system. Sometimes they'll just die. Sometimes they will differentiate. Sometimes they will divide. And sometimes they divide in an uncontrolled way. So we want to understand this process of information transfer. In some cases, like cancer, we want to control it and modify. Right. In some cases, we not only interested in what an individual cell does, but maybe a population of cells, what the population of cells does. How does it respond to the signals and so on. Right, so how do we formulate that? So the idea is that you write this as a channel, maybe, one idea anyway. You have environmental conditions creating signals. These signals stimulate the response within the cell, and this leads to some action, say to fight the virus. How do we measure this? How do we quantify all this? So there are different measures. So one of them is this compact-library divergence, which kind of measures the distance of divergence of the probability distribution of the response given one signal versus another signal. Or this mutual information that we use more, which is a sort of measure of the dependence. I mean, in simple words, there's lots of interpretations, but one of it is that sort of measure of the statistical dependence between the probability distribution of one thing and the other, one thing being the signal and the other one being the response. The challenge, one of the challenges that you face there is that you need to know to compute this. You often need to know the distribution. There are some ways around it, but essentially you need to know the distributions of the signal and the response. And in our case, we have signals and there is possible response, the potential response, they are all the genes. So we have 20,000 genes. It's possible to work out the distribution of 20,000 variables. Right? So we need to do something about this. And one way to approach this, which we use there, is that we stick another arrow here. So we add another output here. So from the response, say the gene expression, we predict what was the signal. So it's like a classifier here producing a prediction there. The benefit is that the mutual information, so this measure of dependence of the signal and the prediction, is much easier to compute, because obviously this is the same dimension like the signal, like 8 conditions, for example, whereas this is 20,000 dimensional. This obviously is much easier to compute. And the other thing is that it's related to the original we wanted, this measure of dependence. So in particular, if in a broad sense, I'm saying this, if you have a classifier and this classifier is accurate, so it's accurate in predicting the signal, then you can cross out the other terms, and this measure that we can easily compute is very close to the actual thing that we want to estimate, the signal to response dependence. So what we do with the data, so we follow fairly standard pre-processing steps, sort of standard because it's not exactly the same type of data, but yeah, fairly standard. And we end up with a table like the one we had, but now the data are normalized. And instead of having 20,000 genes, we have a number of genes, about 2,300 genes. So we filtered a lot of genes, but they didn't seem to have enough variance to be considered to, in our analysis. We just have to throw some away to reduce the dimension somehow. You don't have to, but yeah, it generally helps with dimensionality reductions. Okay, and then the main
Sorry, may I ask a question just about this the identification of the highly variable genes? So you say essentially there is, say, the same gene has the same kind of numbers for each cell, You say, okay, I just discard or not use them in my analysis because there is not that much variability. Is this the kind of identification that you do?
It's a bit more complicated.
Okay, I simplify here.
But I can show you. I can. Yeah, I don't think I can describe, but you find some sort of relation between the variance and the mean of the gene.
Okay.
And you draw some curve with what you expect to be the relation, and then anything above that is kept, and anything below that you throw away.
Okay.
It's sort of yeah, but it's very simplistic what I said now. So right. So then we train a classifier. So we use label data of single cell gene expression and the labels which are the signals to train a classifier to learn the parameters of this classification map. We keep away some data, and then we give this data to the classifier to make predictions. Once it makes these predictions, we can work out things like a classification error and for each signal and so on. And this allows us to estimate the mutual information if we want, or generally study the performance of the classifier. Right, so we've got sort of standard classifications, binary or multi-condition classifications, but also we have this hierarchical structure for the agent, the TNF alpha agent, and we also have these two things, temperature and TNF alpha. So we develop methods for doing this type of classification. The actual classifiers that we use are fairly standard, the usual classifiers of sort of statistical sort of ones. We don't do deep learning or whatever, we do support vector machines. Random forest, that's the most machine learning type thing that we use. Right, so for example, one example of this classification, if we throw these four conditions, temperature and TNF alpha in all different combinations, that's the scores of the penalized LDA, so similar to PCA, but now in terms of focusing on discriminating the different groups. They discriminate quite well. You have overall accuracy very high, so these two conditions are things that we can estimate. We ask, which conditions we can estimate, and we put them in pairs. We have 8 conditions, take all the pairs, and we draw a number of genes against the overall accuracy. You see that they're all on top of each other because the predictions are quite well for most of them. The ones that we don't estimate as well, for example, this one, which is about 80% at the maximum, is the one that you need to distinguish between pulse of every 100 minutes and pulse every 200 minutes. So they're very similar ones as one might expect. Maybe you hope that we can distinguish those perfectly, but it's understandable why you don't distinguish between them. And you have similar things for the others when they're a bit higher. We also combine together conditions. So say put together all the 37 degrees or the 40 degrees and see whether we can distinguish temperature essentially, TNF alpha or not. These two we can do very well. You get 100% precision with few genes already. And then as you go in more detail, then you start making more mistakes. And you see. One thing that strikes me quite a bit is that you have in theory 2,000 genes, 2,000 variables to choose, but still, it's just needing a few. So the whole idea of single cell RNA sequencing, you have all the genome available to measure the gene expression, but you have so much sparsity that you just need a few of them. So it's helpful because now you know which ones to focus on for another experiment, but still, right. Anyway, so that's another discussion. So which features are important? So we kind of try to evaluate which are the genes that they are the most important in different classification, different questions of classification. For example, this 37 unstimulated 40 pulse, and you get a list of the most important genes. You get things that you expect. So these are heat shock genes, so they respond to temperature. We get them to be important in different temperature. These are sort of these things that we expect to respond to TNF alpha. Anyway, another way of seeing the feature importance is this one. So you have sort of heat maps on the back of kappa. So that's sort of the accuracy in some other, different, sort of accuracy measure, how well you're doing with accuracy without going into details. And then you see when you bring one new gene into the model, how much the accuracy improves and so on. Anyway, so I want to get into this bit and talk about the hierarchical thing and so on.
I have a question.
Yeah.
How you compare with just differential expression? Because then you will see the genes that probably just a very small number of genes that are differentially expressed between two conditions, which should be the top ones.
Yeah, I compared them. Boys, I feel that the right thing is the classification because I want to predict what each signal does, not the population. But I guess, yeah, I mean, that's what the margin is.
No, I'm not talking about the population. You do between two conditions. Differential expression is the same as what you're doing, but gene by gene, but using one gene.
I think, yeah, what I'm thinking is if you do a t-test, you're asking whether the mean of one population is different from the mean of another population. I think that's
and the one condition, mean of one condition.
One condition.
That's why you're here too.
Yeah.
With the classifier.
Somehow, yeah, I'm more. You think so? I would love to discuss that.
Instead of a collection of several genes.
Yeah, to me somehow the classifier, because it makes a prediction for each new sample, so it's new cell. It feels to me that it's a bit more, but yeah, it's something that I'm debating all the time.
Expected value of each condition. I think it's the same, but it's fine.
Yeah, no, I'll be happy to discuss it further because I haven't figured it out in my head as well, to be honest. Right, but the results are a bit different, yeah. But yeah, in terms of what genes they come up and so on, not exactly, not completely different, but yeah. Anyway, okay. So coming back to this hierarchical story and so on. So sort of standard classifier for 8 conditions would be looking something like that. So the tree would be okay. At this point, I'm asking the classifier to predict which of those 8 conditions are the ones that generated the data. The hierarchical one will be asking consecutive questions. So you start, say, one sort of hierarchical classification. We'll say, ask first, what is it? No agent or agent? If there is agent, what is continuous or pulse? If it's a pulse treatment, then it's every 200 minutes or every 100 minutes. And then ask the others, the question for the other signal, or the temperature. And okay, so this is much more constrained in how you make the predictions. So I will expect that, what I will expect is that it will give you less accurate predictions under some conditions, but it seems that it's giving us the same kind of accuracy of all this classification of the 8 conditions. And then I said, okay, let me say how it works in terms of prediction. So you have these 8 conditions, 100 cells, say, to make it simple for each condition. And then you ask whether this agent is present or not. If the agent is, it's telling you that in this case, all of the cells with the agent, they were correctly predicted. Those that didn't have the agent, they had 5 errors, which the classifier said that it had the agent there. But then you take all those and you make a new prediction for continuous pulse, the pulse frequency later on, and the temperature. So mistakes that have been made up here, they trigger down there. So it's likely that we'll make more mistakes that you'll get if you ask the question for which of the conditions are there. And it gives you also a nice breakdown of the mutual information and the predictions and so on, so you can understand better, how the gene expression responds to each, in this more detailed questions. How does it identify those more detailed questions? Then I said, okay, so if we can, which is the questions to ask the classifier? We ask what are the labels to choose. So what if I start asking, see when I ask a sensible question, so for example, agent or not, temperature 37 or 40, or say temperature first and then agent or not, what accuracy do I get? And see, it's almost perfect prediction in this case. Compared to something which doesn't really make sense, see how they are split. It says here, for example, at this point, we'd be asking whether it's 37 no or 40 yes. versus 37 yes and 40 no's is mixing up the two things together. Does it give, and then, then you ask the other question. And the accuracy of this is much lower than the other one. And I see that in all combinations that I'm making, for example, low TNF alpha, higher, and so on, asking for the hierarchy things or adding more levels and so on. I always get that the classification accuracy for the sensible ones, they are much better than other ones. Yeah, so see the comparison, all the comparisons getting something back there. So now this is something that I'm still trying to get my head around exactly. But I think one of the things that says the fact that when you're asking sensible questions, you get better classification accuracy from non-sensible questions, tells you at least that the gene expression is affected by these signals, yeah, general thing. Then this hierarchical method is helping me somehow to identify which are the best questions to ask, yeah, to find out what is the signal. So, what are the sensible questions to ask? Yeah, in this case, because of the design, it's obvious what are the sensible questions, but imagine that you have much more complicated signals and you're not sure which are the sensible questions. Maybe this method, the hierarchical classifier, can tell you which are the questions that should be asked from the classifier in order to find out what is the signal. In terms of the biology, what's going on with the biology, I don't know, because of what we want to understand is how the cells make decisions. And in this case, I'm asking, when the cell asks sensible questions, kind of common sense questions, is able to identify, kind of sense the signal much better compared to making some non-sensible questions. So I'm thinking whether we can generate from that a kind of biological hypothesis we can test in the lab to understand how the decision making has been done in the cells. Okay, which I think brings me to the end. So credits to Mike Whiteflat in Manchester that generated this single cell RNA sequencing data. David Ryan at Warwick helped with analysis and ideas here. Yeah, so in summary, we use this high-dimensional data with samples treated on multiple conditions. In our case, the temperature and so on, but think of they could be any kind of interventions. And we want to study the effect of these interventions on individuals and populations. So, yeah, and we use classification and information theory methods to do this analysis. So I think this is a very general description that can fit, in so many other cases. So I'll be very looking forward to talk about these cases, or if you have these cases and you've developed better methodology than what I have here, or if you have different data and you want to see how this performs there, I'll be very keen to talk about it. So I think I'm done. This is some relevant literature. Yeah, thanks for listening.
So just one question about, because this is very interesting, very interesting data.
Thank you.
So what you're saying about that the hierarchical classification works better makes to me makes total sense. Yeah, I've seen that also in other classifiers. And it might be that the right order that gets the best hierarchy is the one that, because cells usually are lazy, they don't want to spend much energy doing things. So it might be that the first classification is the one that uses a smaller number of genes, differentiate between the two, and then you go for more refined changes. So it might be that that is also related with the number of genes involved in each of these conditions. I don't know. Maybe something to check to see if that matches.
It makes sense. There are some genes that they are a few genes actually. Have all these 2,000 genes, but then all your analysis focuses on a few genes at the end, which is there are some genes that they are very good in finding whether the agent is there or not, but also having more detail in them. And then there is some information coming from some other genes that they are more specific to things, but most of the information is on this small number of genes.
So it could be that is the big, the first node, the first branch.
With what you say.
You go there.
Thanks.
If anyone online has a question, please feel free to unmute yourselves ask. Otherwise, I can perhaps ask something for the first part of the talk.
Yeah, sure.
So I'm wondering, I can see how the approximation has worked into, fitting to the data that you have and learning about the parameters. How will it do if you want to predict ahead in the future? Like,
That's no problem because we have, because you have the common fit that produce the predictions and make, you can get the common fit to go to the next step if you want to do it. Or you have the simulations, you can use in terms of the simulations. If you don't have data, you can still make predictions in the future. Because I mean, the whole point of it, I suppose, is that you learn the mechanism, what is going on actually in the cell, or at least under your assumptions. So then predictions are very easy to, very cheap to make because you learn the whole system. So that works fine in that.
But this is all depends on your normal approximation.
Yep.
And if you can make the approximation, what do you do?
If you can't make it, not many things. So there are other approximations, but I don't think for these large systems you can use them. What I'm working on is to generalize this. So instead of having oscillations, if the dynamics are linear, then it works in the
Usually they're not.
But if they're non-linear, then what I'm studying is see what kind of dynamics you have, and then build an algorithm which follows this idea with the corrections and so on, to make it, which doesn't slow down the whole thing a lot, but still proves a lot in the accuracy, and so it's working. So
in terms of the algorithm in the first part, like the three algorithms that you showed, like are for distinct purpose distributions, right? Like I would expect the tempering to work in a multimodal distribution. And you said it's mostly flat. Or
so would be mostly flat for those that we have low sensitivity. Right. So the ones we showed, the multi-modes and so on, those are ones that we have high sensitivity. I'm not sure what happens. If you add a lot of parameters, we have low sensitivity. If we see multiple modes, or that's something that's not too sure about. Should look at it.
Can you look for pairs of parameters? Because
we include up to 10 parameters, in some cases 20 parameters with, you don't get the same kind of result because of the low sensitivity. But we tried that. It works with some success. You estimate the parameters that they have higher sensitivity, and estimate less well those that they have lower sensitivity.
Are there any more questions? If not, we can thank you again. Everyone online, in person for attending, and I'll see you.
Thank you very much. Rush.